Thank you. Thank you. So good evening. Thank you for still being there that late in the evening. The good news is this is not going to be a lecture like the other ones where we really want to go deep into a technique and learn everything about it. So the idea is really more to give you a little bit a relaxing end of the evening where we show you a little bit what you can do with the techniques that you have learned so far and that you're going to learn in the next lectures when they apply to a certain scientific field. So in our case, it's going to be the field of heritage research. Now, since the lecture is going to be put on the internet, it's also important to note that is, of course, not my own work that I'm presenting here, but the work of colleagues of mine that I've selected for different pedagogic reasons. So the legal stuff aside now, what we're going to talk about is first I'm going to give you a short introduction. Yeah, it was really interesting. So we're going to talk about what is heritage science actually, so what are the samples that we're looking at typically, what are the scientific questions we want to answer, and then we're going to make a small cross-section through the different techniques that are used. Well, only the most common techniques that are used in cultural heritage, because as you can imagine, almost all techniques had synchrotrons. Sorry? Oh, sorry. Yeah. So as you can imagine, all techniques that have been that are in large-scale facilities that have at some point been used for cultural heritage sciences. So we're only really going to look at the most commonly used in this short hour that we have. So first of all, what is cultural heritage science? So it's of course a good question. And so we consider cultural heritage science everything that is done in the scientific communities of paleontology, archaeology, conservation science, and the sciences of museum objects. And now we're going to have a look a little bit more in detail at each of these different fields. So let's start with archaeology. It's probably the one that you're thinking of first when you're thinking about cultural heritage research. And here we have really a large variety of different types of samples that we typically encounter. So it can be from ceramics, of course. I don't think I'm going to use the pointer that much, actually. So ceramics, of course, is something probably you think really quickly when you think about archaeology. So pottery, of course, but pottery can also be, or ceramics can also be tiles, like in architecture for doing floor or for doing roof tiles. Then we have pigments. Pigments, of course, a really large field for starting from the first prehistorical grottos, where we have like cave paintings, when all the way up until today, where we can have the different forms of pigments. Then metals is, of course, a very large class, when we can have tools and we can have weapons and armor and things like that. Glasses, so in ancient times, glasses are mostly opaque and used for jewelry. And then later we have transparent glasses for vessels, for example. Now textile is, of course, a really important class of objects, but since they are organic, there's very few that actually from ancient times that are conserved until today, or only in very particular conditions. And lastly, we can have human remains like mummies and hair and so on that can also be analyzed to give us some information about its origin. And so the questions that we're typically asking are the questions of provenance. So where did an artifact come from? So where has it been produced? Where did the raw materials come from? With which cultures has it been traded to? Because this is, of course, very important to understand which cultures had some technical or some trading network and thereby were sharing knowledge and culture. Then we have the manufacturing process. We want to understand how a certain artifact has been constructed. Again, this gives us more information about the knowledge, the science, and the technology that was available to a certain culture. And lastly, I hope you can see this down there, we want to understand the aging and corrosion processes. So we have an artifact that has been lying in the soil or in seawater, for example, for thousands of years or for hundreds of years, and we want to understand what was its original state. So the next big field is paleontology. So here, in contrast, we have a very clear set of samples. So we're looking at extinct organisms that come to us as fossils. So typically fossils in stone, but also fossils in amber. One particularly interesting category is teeth, because teeth allow us to get a lot of information about the organism. So I don't know if you know, but teeth also have an effect like growth lines. So every year you get very fine line in the teeth. And it means if you find a fossil with its teeth, you can actually understand how old this animal was before it died, or at least before it lost its teeth. And also, So if you have like certain special events in the life of a creature, or like a special diet at some point, this can be can leave traces in the teeth, and so it can get a lot of very interesting information. So the questions we're typically asking in paleontology are the question of evolutionary dependencies. So where in the evolutionary tree does a certain fossil fit? Which are the creatures it depends from, and which are the creatures that depend from it? Then we want to understand the functions and behavior. So basically, how did this creature live? What did it eat? And what was the environment like in which it lived? And lastly, we want to understand the process of fossilization. So the question, how did a dead animal or a plant transform over thousands of years into an anorganic system? And what are the chemical and biological markers that are preserved in this process that we can still use today to learn something about these creatures? So the last two points I put together because they're looking at the same samples. So this is conservation science and a science about museum objects. And here we're looking at everything that you can find in a museum. And you know, of course, in a museum you can find everything. So from the first thing, of course, you probably think about the artworks, like in an art gallery. But you can have like everything. You can have like monuments, like castles or churches. And like in a moment, a big question conservation science is Notre Dame de Paris. That after the fire, of course, has to be reconstructed. And there's a lot of research going on to understand what's the best way to reconstruct it and to conserve, let's say, the structure is still there, but has been, of course, severely damaged by the events. But we can also have like non-decorative objects, like technical objects, like military objects, like vehicles, like airplanes, things like that. Basically everything in a museum. So this is a large set of samples, potential samples, but the questions that we're looking at are comparably clear. So in conservation sciences, we want to understand degradation processes. So that means we typically have an artifact, for example, a painting or something, that we know has been degraded either before it came to the museum or during the lifetime it was at the museum. And we want to understand why is this happening, and can we do something to slow it down or to prevent it in other artworks and things like that. And of course also if we already now have a changed artwork, we want to know what did it look in the beginning, what did it look like when the artist really painted it. And the other point is the science of museums objects. So this really depends, of course, on the type of museum. So in an art gallery, we are asking questions typically about art history. For example, what techniques did a certain painter use? What kind of pigments did he use? In which way did he apply them on the canvas? Or if you have, for example, a military museum, then you have questions of military history. So in which order were certain weapons developed? What kind of materials were used and things like that. And sometimes you can also detect either frauds or also parts of an object have been replaced over time. For example, they were damaged and had to be replaced. This can be also important to better understand the systems with which we are working. Now, I've shown you it's a very, very diverse set of potential samples. And you can ask, of course, the question, well, is it actually useful then to group all this together in one term under heritage science? And the reason why we think that's useful is that while they're very different, they have things in common that they don't have in common with other branches of science typically. And this is that we have limited a priori information. So when you're doing an experiment, for example, in material science, then you're preparing your sample for the experiment. And you're going to do it, of course, as pure as possible, optimized for this experiment, and you know exactly what you're going to put into the beam. However, when we are doing heritage science, and often we're taking a sample that has been in the soil for a long time, we know roughly what it is. Of course, we pre-characterize it before going to the beam, and we look at it in a microscope, things like that. But in the end, you don't really know what you're going to find until you do the experiment. And the other problem is that we have often very highly heterogeneous samples. So often they're complex just from the beginning, because you have, for example, paint samples. So you have a little paint layer where there are pigments in a binding media, and then you have different layers. Or that's like organic samples, like wooden samples that are intrinsically very heterogeneous. But even comparably simple samples like a metal object, a metal tool, for example, you could say it's just a block of metal. But in our case, we often then have corrosion layers. We have many impurities inside, because, of course, in ancient times, the raw materials were not as pure as they are nowadays. And so this is why we are going to use mainly imaging techniques to overcome these points mainly. Okay, so let's have a look at the different imaging techniques. As I said, only the ones that are the most commonly used. And the most common, we're going to start at a synchrotron, is basically hard X-ray microspectral imaging. Yeah? So what is this? So it's basically everything we use, like focusing optics to focalize your beam on the sample. Then you're doing a certain type of experiment, for example, a spectroscopy. And you get this information that you're looking for this point that is in the resolution of your beam size. And then you're moving your sample, and you take another experiment at the next point, and then you can reconstruct an image of your sample using this information you've obtained. And the most commonly used and most straightforward maybe system or technique to use is X-ray fluorescence spectroscopy. So just to remind, this gives you the elemental contrast of your object. And it's really one of the most popular techniques in cultural heritage research because with elemental contrast, you can do a lot of interesting things already. Yeah? So this is so interesting that it's also used with like mobile instrumentation, so that like hand pistols basically you can use at an archaeological excavation, for example, to just shoot at objects and you get a spectra. Yeah? And there's also semi-mobile instrumentation you can transport, for example, to a monument where you would look at a wall painting that obviously you cannot transport away. And nowadays, most big museums, they have lab instruments, so lab scanners that they can use to scan a complete painting, for example, with X-ray fluorescence. So they don't need to take their own objects and take them out of the museum, which is, of course, for them a big advantage. So people generally only come to the synchrotron when they really need the synchrotron, and it's typically for having very high access to very high spatial resolution. And or if you want to look at trace elements, for example, because there you need a very high flux. Okay, so the first example I want to show you is not something really spectacular, but it's something that is really like the bread and butter of most of the technique. It's not something you're going to find in the newspapers, but it's really, let's say, the groundwork, the majority of the work that is done in cultural heritage. Huh? And this is using X-ray fluorescence as a help for provenancing. That means for finding out where an artifact comes from. And so X-ray fluorescence spectroscopy can be very helpful there because we can look at the trace metal contents of a sample. And the trace metals are often very particular for the region where a certain sample has been produced. Because, for example, the mines, where the raw materials that have been taken for producing the sample, they have a very specific profile of trace metals. And then if you look at a corpus of samples that you found, for example, in excavation, you can look at the trace metals and you're going to see, for example, oh, there's a certain part that has this profile and other has this profile. So these are probably from different regions. So the example I want to show you is about the provenancing of Greek artifacts. So this is a set of artifacts that has been found during excavations in the Sicilian city of Gela. And so when they did these excavations, they found out that their corpus came out from 3 different production centers. So they found pottery from Attic productions, so around Athens. They found local productions that were made in Sicily, and they found productions from other Greek colonies in the south of Italy. And these provenancing or the decisions where this probably comes from is done by a static comparison. So the archaeologist looks at the sample and says, oh yeah, this is very typical for an Attic production because it's a very strong gloss or something like that. So here you can see some examples of the different productions. So you have for each production center you have one example, and you can see for the untrained eye it's not so obvious to do this provenancing. So you can maybe see that this is maybe a bit darker than the others or so, but as you can imagine, especially if you have only a fragment left, it gets very hard to do this. And so the question was, can we give some tool to the archaeologist to help him in the decision where the different artifacts come from? And so the idea was, let's look at the trace metals both in the clay and in the black gloss. However, the problem is that the black gloss is very thin. It's typically around 10 to 20 micrometers thin. So you really need a synchrotron here to be sufficiently resolved spatially to see really the difference between the glaze and the clay. So this is a photo of the experimental setup. And so you can see here on the left, we have our focusing optics. This is in this case a KB mirror system. Then we have our sample here. And so we're now going to illuminate the sample with a microbeam with a spot size of a little bit less than 10 micrometers. And then, of course, the sample is going to submit fluorescence radiation in all directions. So we're going to take this with the silicon drift detector, and then we're just going to move the sample through the beam so that we can obtain an image with the elemental distribution. So at every pixel we take a complete spectrum, and it means we get all the all the elemental contents in this spot that are accessible to our energy. Yeah? And just show you a few examples here. So just for some elements where we saw something interesting. So you can see that the lower part here is the clay, this orange clay. And here the red part is the red part here is the black gloss, and on top of that we have air. Yeah? And so you can see the gloss is iron-based, so that was known, of course, before, but you can see that we separated very well from the clay. And then you see in other elemental distribution, you see other interesting features. For example, in a strontium, we can see the subsurface layer here. Yeah? And this is very interesting for the archaeologist because it shows you that it's a relic of the fabrication process. Since it's below the gloss, it's probably a step that has been taken before the gloss has been applied. And the same is with the zinc. So you can see this is only present in some of the samples. So this is again interesting to see which fabrication regions used certain techniques and which not. But you can also see it's not homogeneous as the iron is. So it seems to be something that has been applied at the very end after the gloss has been done and then diffused into it. So this could be, for example, another step that has been taken. So it's known that sometimes these potteries were treated by acids as a last step. And this could be a remaining of this. In addition, so we get the trace metal contents here, and you can see for the different productions we get some differences, especially in the gloss. And this is now very helpful for the archaeologists because with their mobile instrumentation they can, for example, look at the paste without problem. They can see what trace metals they have there, and if they are still in doubt, they can look directly on the glaze, and this gives them also an idea about the trace metals inside and can then help them in finding and deciding from where probably their artifact came from. So as I said, this is really a typical archaeological study. The next example I want to show is the contrary. It's a really spectacular study. I included it here because it was historically very important. It was basically one of the first synchrotron studies of a cultural object that really went viral, I would say, nowadays. And it really started a little bit this trend of doing X-ray fluorescence on paintings. And it was really because of that the museums then were interested in having scanners for them at their museum to investigate their paintings. So the painting question is called A Patch of Grass. As you can see, it's aptly named because it shows you a patch of grass. It has been done by Vincent van Gogh in 1887 during his years in Paris, and it's nowadays in the Kröller-Müller Museum in the Netherlands. And when it arrived in the museum, they did, of course, some analytical techniques on it. And one of the things they did was X-ray radiography. And it's not very well visible, but you can see there's some substructure that clearly has nothing to do with the painting you can see in the front. So it was very likely there's a painting underneath, which is not an enormous surprise. You maybe know that artists often overpainted paintings that they've done before, which they were not very happy because canvases were very expensive and Van Gogh was not a rich man, so it was not a surprise that he did this. And of course, if it's an artist that is so famous as Van Gogh, you really want to know more about this hidden painting. So the question was, can we learn more about it? And for this, they went to the synchrotron, went to the now not working anymore DESI ring, Doris ring at DESI. And they did X-ray fluorescence analysis. I only show you here 4 of the maps of roughly the region that was only also shown in the radiography. And you see already in the antimony signal, you see a very good, here. You see that the face is much clearer visible than it was in the radiography. So already this is, of course, quite a spectacular result. You can much better see what we had before. That would have been already worth going to synchrotron. However, that's not everything we can do, because now we can use these elemental maps and try to correlate it with the pigments that Van Gogh used. And luckily, Van Gogh, he liked really experimenting with pigments, and he wrote about his experiments to his family members in his letters. So we have a really good idea what kind of pigments Van Gogh used in his works. And so we can try to reproduce the colors he used. So lead is most probably lead white. It's a pigment that has been really, really common over most of history. Actually, it was basically the only white pigment that was available until the 19th century, where semiconductor pigments became available. And you can see it's very, very homogeneous. So this is most probably the grounding layer. So next we have Naples yellow. That is a pigment that contains antimony. And it's called Naples yellow, obviously, because it's a yellow pigment. And as you can see, it's mostly in the flesh tones. So that would make sense. Another very famous painting is pigment is vermilion, which is a mercury-based pigment. It's a very bright red. And lastly, we have zinc. So that was a quite new pigment. It's zinc white, the zinc oxide. That was quite new when Van Gogh lived. And you can see he mainly used it here for the highlights. And now with this information, we can try to fuse this information together to get an idea what the original painting looked like. And of course, there's a lot of guesswork when you're doing this. But it gives you an idea what it could have been. And of course, this is now interesting for the art historians because it gives them another step to judge how Van Gogh's style evolved over the years. And you can see here other paintings that he has done about women faces. And you can see that, for example, the one here on the top, of course, is very similar to the one that he had overpainted. So this is, of course, if you're really following the development of Van Gogh's style, this is a very interesting result. OK, let's go to the next technique. So this is X-ray absorption spectroscopy. Well, you just heard everything about it, so I'm not going to go into any details here. Just going to tell you that in cultural heritage, we mainly use Xanes. There's less EXAFs, it happens, but it's more or less common. It's a very, very popular technique in conservation science, because in conservation science, we're typically interested in following chemical reactions that are taking place over long time scales. And so we want to know what is the oxidation state, for example, of our element. And this is also the example I want to show you is from conservation sciences. So we're going to have a look at the degradation of smalt. Smalt is a very, very popular pigment all over basically all over time, so you can still buy it today. It's a potassium silicate glass that is colored blue with cobalt oxide and then ground to a very fine powder, and then you use some binding media like oil and you put it on your canvas. And it's comparably cheap, it's easy to do, and it was very, very popular, even though people knew quite early on that it's not a very stable pigment. And it has this tendency to degrade into grayish, brownish tones. And indeed, if you go to art galleries, you're often going to find paintings that have this grayish, brownish skies and things like that. This is typically a degradation due to this process. And you can see this, for example, in this painting called The Heavenly and Earthly Trinities, where the sky has been painted in blue. And you can see nowadays it's a brownish, grayish tone. That doesn't at all transpire the beauty of its original. And you can really see it's a problem with the smalt. For example, you see the texture here that Maria is wearing is still blue because it's another blue pigment that was being used for that. So the question is, what is happening here? Why are these pigments degrading? And to answer this question, the scientists took a little sample from this image, and they looked at it under the microscope. So you can see a cross-section here. So you have the different paint layers like this, well, parallel. And so you can see this is basically the outermost layer. And you can see clearly the different glass particles of the smalt. And you can see some particles are still very healthy. They are nice and blue, while others are degraded and are grayish, as you can see here. And now using a microbeam at a synchrotron, the scientists were able to take X-ray absorption spectroscopy or spectrograms from the well-preserved and the altered pigments and compare them. And this you can see here. This has been done at the Lucia beamline at the synchrotron Soleil. And you can see in black the spectrum from a well-preserved smalt, and in red you can see an altered smalt, a grayish smalt. And you can see they're very different. Now I'm going to skip over the whole data analysis. I'm just going to show you the conclusion. So what the scientists found out was that in the original blue smalt, the cobalt is in a tetragonal environment. So basically the oxygen atoms around it are forming a tetrahedron. And it's known that this configuration is not very stable. In fact, it's only stable in the presence of potassium ions. As I told you, smalt is a potassium silicate glass. And what's happening now is that the binding medium that has been used to put the glass on the painting is actually leaching out these ions over time and leading to an instability of this configuration, and it's decaying into an octahedral configuration in which the cobalt has this brownish grayish color. This is, of course, now the first step only of for the conservators, and now they know what is the chemical reaction that is happening, and now they have to look at what are the environmental conditions, for example, that are favoring this. So how can we better protect our artworks? Which artworks are especially maybe prone to this kind of change? And so that we can hopefully in the future better preserve our artworks. So the last X-ray technique, 2D X-ray technique I want to show you is X-ray diffraction. Now, X-ray diffraction in cultural heritage is mainly used to determine the phases that are present in our sample, so the mineralogic phases. And this can be used to identify the pigments that we have in our sample, and it can be used to shed some light on the thermal history of the sample. For example, if we have ceramics that are typically heated to very high temperature, if we find some high temperature phases in our sample, then we can judge that we probably heated our sample at some point to this degree. So the example I want to show you is again about pigments. So here we're going to look about lead white. As I told you, lead white was really a very, very common pigment. And it's a white powder that is produced by corroding metallic lead. And you do this with the help of acidic vapor and with CO2-rich conditions. And there were lots of different recipes that varied a lot from region to region and over the different time periods. And they all have in common that you end up with a white powder, which is a mixture of hydrocerusside and cerusside. You can see the chemical formulas here. And the ratio between these two crystals depends really on the fabrication process. And they have slightly different crystalline sizes and so on, so they have slightly different properties. Now this study became very interesting when scientists in the Netherlands started to look more closely at Rembrandts paintings. So there was a big campaign to look at different paintings from Rembrandts and to investigate different techniques that he used. And one very famous technique that Rembrandt used was his impasto layers. So these are layers where he put really a lot of paint on the canvas, giving it really a three-dimensional structure and thereby some peculiar optical properties. And the question was really to better understand why and how he did this. And you can see an example of this technique here in this portrait of Martin Solmans that was painted 1634 and is nowadays in the Rijksmuseum. And you can see it's used here on these decorations that he's wearing. So you can see here a more close-up look to these flowers. And you can see here this big droplets of paint that have been put on the canvas. And so the scientists took different samples from different pictures. So we are going to have a closer look at a sample taken from this picture called Bathsheba. It was painted 1654 and is nowadays in the Louvre. And you can see a photo of the cross sections up here. And you can see clearly we see two very distinct layers of lead white. So if you do X-ray fluorescence here, you're going to see it's both lead white. And well, that alone is maybe not a big surprise, because if you want to really have a three-dimensional structure, it's not surprising that you maybe go in different steps. But what was a big surprise was looking at the diffractograms. So this is work that has been done at ID13 at the ESRF. And you can see the result here. So this is an image which every pixel, a full diffraction image was taken, then analyzed, and the contents of hydrocerusside and cerusside and a third phase that I cannot go into detail now for time reasons have been calculated and plotted here. So in green you see the hydrocerusside concentration, in blue the cerusside concentration, and you see clearly that the two paint layers are completely different. So it's not just he used twice the same paint and he just made two different layers. He used two completely different types of lead white, and it's something was completely unknown before. And this is now very interesting, and you can also see that this has been done not just by accident. He ran out of paint and he just used the next batch. He has really done this because he wanted to use the different optical properties of a lead white mixture with mud, with a large content of hydrocerusside and one with a low content of hydrocerusside. So that was really, as I said, very interesting for the art historians and really showed them that at that time already it was known basically that the optical properties of the powder were different for different recipes, and we could afterwards also see that actually he bought different types of lead white that were also differently priced, so probably the production method is actually a little bit different for both. Okay. So this is we're going to leave the hard X-rays for the moment, and we're going to go to the opposite spectrum end of the synchrotron spectrum, to infrared. So infrared you can use to do Fourier transform spectroscopy. And so this is a spectroscopic method where you shine an infrared beam on your sample, and then you're going to excite some of the vibrational bands of your compounds in the sample, and you're going to measure an absorption where there. And this is very helpful because you can both probe organic and inorganic compounds with this, which is very helpful because we often have both in our samples. And of course, now you can say, but FTIR, I can do it in my lab, why would I go to the synchrotron? Indeed, the advantage of the synchrotron is, first of all, that you have a very large spectral range that you can scan that is very flat. And the second advantage is that you have a very small source size, and it makes it easy to focalize your infrared beam on a very small spot close to the diffraction limit, which is around a micrometer for infrared photons. So the example I want to show you has been done on musical instruments. So it was a study that was done with the Musée de la Musique in Paris, and the scientists wanted to look deeper into the varnish of these instruments. So you have probably heard of the famous Italian instrument maker Antonio Stradivari. So his instruments were known to be some of the best that have ever been made for their acoustic properties, but also for their beauty. And one peculiar thing about them is this beautiful reddish tint that they have. And for generations, people have wondered how he managed to do that, if he had some sort of special ingredient, special recipe that nobody else had to achieve this beautiful color. And to know more about this, the scientists again took a little sample of the wood here of the violin, of several violins actually, and they looked at it under the microscope, so you can see the effect here. So you can see we have the wood at the bottom. Yeah, you can clearly see the wood cells, and then you have two distinct varnish layers, a lower layer that is colorless, and then an upper layer that is slightly tinted. And now, thanks to the high resolution of synchrotron infrared spectroscopy, we can look at each of these compounds in detail. And you can see here the spectrums that have been taken from the different parts. So we're not going to look about the wood anymore. But you can see the both layers on the top, they are very similar. And we're going to look a little bit what we can find. So first of all, you see that there are not many features you can see. You see the two bands that are already highlighted here. So these are typical for carbohydrates. So these can be attributed to partially hydrolyzed and oxidized drying oils. So these are really things that are really typically used in woodworking. So this is nothing special. And then you see, oh, well, I don't really see much else actually. What's interesting is what you don't see. We don't see, for example, amide bands. So that would be here. This would be typical for proteinic material. So if like egg or casein would have been used, you would see a signal there. You also don't see any signal here. It would be typically for gums or waxes. So the scientists use a lot of other techniques that I'm not going to detail here out of time reasons. But in the end, they came up with basically nothing. So there's not a single really unusual ingredient that they could find. And so they concluded just a silicative oil was applied as a sealant. And then a second oil resin layer was applied. That this is a slightly tinted layer that you have seen in the microscope images. And absolutely nothing spectacular or unusual has been found. And I really like the conclusion of this work that said that even if it's a little bit frustrating that after using all these techniques, we haven't found anything unusual or spectacular or magic in it, the nice conclusion is that the beauty of this instrument is just due to the extraordinary skill of the craftsman in the end. And I think that's a really nice conclusion actually to a study that actually showed had no positive result in the sense. Okay, so now we're going to go over to neutrons. And one of the very much used techniques for cultural heritage is prompt gamma ray activation imaging. So this is a little bit the neutron equivalent of X-ray fluorescence spectroscopy. So what you're doing here is you use a cold neutron beam, and you illuminate your sample with that. Then the nuclei in your sample are going to capture the neutrons. They're going to become activated, and they're going to decay by emitting gamma rays. And these gamma rays are characteristical for the isotopes. And this way, by measuring the gamma rays, you can understand what kind of elements you have in your sample. So the setup is like you can see here. So you have a cold neutron source. You illuminate your sample, and you look at the gamma rays with a gamma detector. Now, you can do this also in imaging mode. by slitting down the beam here and then putting a lead collimator in front of your detector. And now you know that all the photons that you're going to collect are from this volume element here in the center. And then you just scan your sample through the beam, and so you can get a 2D or 3D analysis of your sample. So here you can see a PGA setup at the research reactor in Munich. Unfortunately, you cannot see much. So you can just see here the neutron flight tube. Then there's a lot of lead shielding, and then here is the gamma ray detector. And here's an example that has been measured by this technique. So it's a medieval brooch. You can see it's comparably large. And you can see here a neutron tomogram that has been done of the brooch. And then you can see here the different elemental compositions. And you see the big advantage of doing this with neutrons is that you can get at the same time high Z elements like gold and silver as low Z elements like sulfur and hydrogen. Yeah? And you can penetrate the whole sample, and you can get information from the whole sample, even though it's made of really absorbing material like gold and silver. Another very important neutron technique is diffraction. Yeah? So this is, of course, a lot like X-ray diffraction. The big difference is that in neutron diffraction, you can make use of a certain trick that the neutrons, the speed of the neutrons depends on their energy, yeah? While the speed of photons is always the speed of light. Yeah? So that means if you can measure the flight time of a neutron, you can reconstruct its energy, and it means you don't need to monochromatize your beam, so you can use much more flux. So the setup looks like this. So you either use a already pulsed source like a spallation source, or you use a continuous source like a reactor with choppers, so that you know when the neutrons arrive in your beamline. So they come to the sample and get diffracted, and then in your detector, you just measure at every point the position and the arrival time of the neutrons, and thereby you get the angle and the energy of the neutrons. And this way you can reconstruct your diffractogram, and then you can, for example, do phase analysis just as with X-rays. So with neutrons, you have the advantage you can look at larger objects because you can penetrate typically larger objects, but typically with less resolution. However, you can also do something that's very hard to do with X-rays, and that is texture analysis. So I don't know if you know what texture is. Texture is a state a little bit between a perfect single crystal, where all the crystallographic planes are perfectly aligned, and a perfect powder where you have little crystallites that are completely random. So in this last case, you would get like Debye-Scherrer rings. And in the case of a textured sample, we have that there are grains, but they have a sort of pre-alignment. They're not completely random. And this is going to give rise to incomplete rings. And this is a very useful technique because many fabrication techniques, especially in metal, are going to leave certain imprints on the grain distribution. And then by measuring the texture of this material, I can deduce how this material was fabricated. The disadvantage is I need to penetrate the whole sample to do this, and most metals, of course, are very absorbing to X-rays, so that's just why this is really more done with neutrons. Now, the texture of an object is typically shown in pole figures, so this is a little bit a concept that is a bit weird when you're seeing it for the first time. It's actually quite simple. So what you have to understand is that a pole figure wants to show you the relationship of a certain set of crystallographic planes. For example, if you have an iron sample, the iron 111 planes, in respect to a certain direction of your sample. For example, if we take a cylinder as a sample, we want to say what is the orientation of the iron 111 planes in respect to the top of my cylinder. And then I just imagine that I have a circle on top of the direction I want to look at, and now I'm going to look where the normal vector of my crystallographic planes is going to penetrate this circle. So for example, if they are all perfectly parallel to the sample surface, then the normal vector is going to be exactly in the center, and I'm just going to get 1 point in the center of my circle. Now if they are tilted a little bit, let's say they would be all tilted in this direction, I would get a point here. The same if they would be tilted in the other direction, I would get a point here. Now you can imagine a more realistic system, you're going to have that they are maybe all tilted in a certain angle, but still in random order, and then of course you would get a ring. And this is interesting because this is, for example, a typical texture that you obtain when you're hammering a material, a metal. So if you have like a metal block and you're hammering it to shape, you're typically going to end up with this type of grain distribution. So let's look at an example. So the example I want to show you is, has been done about silver coins, so more specifically about silver thalers from the Kunsthistorische Museum in Vienna. And the scientists here wanted to follow the development of coin minting technology in the 16th century. And so we're going to start with coins from the early 16th century. And here we're going to get this type of textures. So you can see we have here the orientation of certain silver planes, so the 1,1 plane, the 2,0,0 plane, the 2,2,0 plane, in respect to the surface of the coin. And you can see here we get rings. So this is typically what I showed you before, the typical signature of a hammered sample. So it means in the early 16th century, they used their substrate, so the silver cylinder, and then they used a stamp they put on top, and then they hammered the stamp into the coin to produce basically the image. And now if we look at coins from the later part of the 16th century, we see that the texture patterns look completely different. And these now are the typical texture patterns of an object has been rolled. That means now you have a cylinder where there is this image you want to engrave is embossed in it, and you roll this over the coin. And what's interesting, we can even now say which is the direction in which it was rolled, because it's perpendicular to the stripes you can see here. So even now, more than 400 years after these coins were fabricated, we can still say in which direction the cylinder was rolled over the coin. I find that comparably fascinating. The last part, was that there are some samples that they found in this study that looked a little bit different. They looked like this. This is irregular. And this is more typical actually for samples that have been solidified from a melt, so cast basically. And this is not so good because it is typically what is used to make forgeries. So if you want to forge something, if you want to take a duplicate for something, what you typically do is you take the object you want to duplicate and you press it into a soft support, like a wax, to get its negative image. And then you fill that wax with liquid metal. And you let it solidify, and voila, you have your copy. And this is why most forgeries are actually cast, and you can see they leave this strange, incomplete texture patterns. And so it was advised to the conservators of this museum to check if these might really be forgeries or not. Okay, so the last part of our talk, we're going to look at 3D imaging techniques. So there are different ways of doing 3D imaging. For us, basically, it's just one that is really important, that is really commonly done, and it is computed microtomography. This works really similar for X-rays and for neutrons. So what you basically do is you take your sample, you illuminate it with a beam that is typically larger than your sample, or at least it's larger than the area of interest that you want to reconstruct. And then you just put a detector behind it, and you take a radiograph, and that gives you the 2D structure of the sample. And now if you turn the sample and you retake a radiograph, you get more and more information about the 3D structure, of course, of your sample, just by turning it. And then you feed all of this into a clever algorithm, and then here it reconstructs your complete 3D structure. And this can be done with very, very high precision. Of course, then many projections are needed. So in a typical synchrotron experiment, you're going to use thousands of projections, and then you're going to recombine them into a volume that is very, very large, but very detailed and completely describes your 3D structure inside the sample. So you can imagine it's a very powerful tool for many different cases, and it's used in many different systems in cultural heritage research. However, it has become really a fundamental column in paleontology, because for paleontology, you can really now see with very high resolution the complete reconstruction of your fossil, not only the visible part, but also the part that is hidden in the stone and the internal features. And so most big paleontology laboratories have indeed now bought their own lab scanners to do laboratory-based tomography to do this. However, this works only for some fossils, because the main problem that you have is, contrary to medical imaging, for example, where you say, okay, I want to see the radiograph of my hand, so I'm going to see the bones because they absorb much more than the skin or the flesh. The problem with fossils is that you have a rocky fossil in a rock matrix, so you're not going to have any absorption contrast. So you have to find, for most fossils, or at least for the most interesting features in fossils, you have to find a method that is more precise for these type of systems than absorption contrast. And the method can be used here is phase contrast tomography, and this can typically only be done at synchrotron sources. And what you're using here is not the absorption of your sample, but you're using another property. Because you have such a brilliant source, such a small source, such a high quality source, you know that the wavefront that is going to arrive at your sample is very well defined. And this wavefront is now going to be disturbed by its passage through the sample, and especially by the interfaces inside the sample. And then there are different techniques to make this visible. The most commonly used technique is actually the most easy technique. It's called inline phase contrast. You can see it's very simple. So it's basically the same setup as we had for absorption contrast, only now we use a high brilliance source, like a synchrotron. And you can see we have put the detector a little bit back. So we have put him at a certain propagation distance from the sample. And the effect of this is that now the wavefront that has been distorted by the sample has some time to propagate, and we're going to see some intensity variations that are going to appear at the detector as a function of the distance. And this is shown here in this simulation that has been done by a colleague of mine. And this is a simulation of growth line in teeth. So you can see a material represents growth line in teeth. So you can imagine something that's is very thin and has very low absorption contrast. And in fact, if you put your detector directly behind the samples, you cannot see anything. You see there's no absorption contrast at all. But now as you start to go further away, you can see there's some contrast appearing. So this is really the distorted wave field is starting to develop intensity variations. And as further as you go away from the sample, the stronger this becomes until these variations start to overlap. And then, of course, it becomes difficult to interpret them. So the importance about this technique is really that you find the good distance between sample and detector, which depends on the energy that you're using and typically on the resolution that you want to have for your sample. And you can get with this technique really very impressive results. So I just show you a short picture here. So this is a tomogram that has been taken about a fossil fish. So you can see the skull of the fish here. This is actually a very important fish because it's actually the first specimen of fish that had jaws. And as you can imagine, it's a really important knot in the evolutionary tree because, and of course, from this, all the mammals and all the other fish with jaws depend. And so it has been observed with phase contrast tomography. And you can see here that you get it with very high resolution, but also you can image really here, you can see veins and arteries of the fossil and the nervous system. It's, of course, really incredible. You would have never thought, I guess, that you would have seen this in this old bone, right? However, the study that I want to show in a bit more detail is about another fossil. It's also a very famous fossil, Archaeopteryx, that you've probably heard about. So it's a fossil, an extinct creature that was very important for, of course, the theory of evolution, because it's clearly in between the type of creatures we now nowadays as dinosaurs and the creatures that we call nowadays birds. So this was very important for the development of evolution theory. But it's also one of the questions that's always wondered the scientists is, was he actually able to fly? So it's a creature that still has lots of the properties and morphology of a dinosaur, but we know it has wings. So the question is, could it fly or could it only glide? And for the most time, people thought it was only able to glide because it doesn't have the morphology of a bird. So people thought it was just climbing up trees and then going from one tree to the next in gliding fashion. However, this is, of course, not the end. So some people thought, let's have a look in more detail. And they wanted to examine the bone structure of the forearms, basically, of Archaeopteryx and compare it with other extinct and living creatures of which we know exactly how they behaved. And the problem is, of course, now a fossil of Archaeopteryx, you cannot just cut into it and look what it looks like, because, of course, they are very, very precious. And for this reason, they needed to do high-resolution tomography and then do virtual cross-sectioning. And you can see this here. So they focused their study on two bones, the humerus and the ulna bones. So these are these bones, so this one and the ulna. Because these are the ones that are most modified for the different specializations in movement. So you can see here a virtual reconstruction of these bones of one specimen. And you can see here that these colored lines, these colored lines where virtual cross-sections were taken. And you can see on the second picture a virtual cross-section that has been extracted. You can see, of course, that it has been smashed by the process of fossilization. And so the scientists puzzled it together again, so they get the picture of the right, which is like the bones were in the still living animal before they were pressed together. And now you can look in this with more detail. So one thing they noted was that the cortical vascular density is very similar to the ones of modern birds. There's already a first step that tells us that there are probably Archaeopteryx did much more steps towards modern bird flight than is obvious from his morphology. So cortical vascular density means basically the number of blood vessels that you have around the bones. They also saw the cortical thickness, so the thickness of the bones is thinner than in non-flying animals. Another point that probably were closer to actually flying animals. And then they did other techniques where they look at different parameters of the bones and they plot them on top of each other. And this makes the different types of creatures cluster together. So I cannot go into detail now what is actually the parameters that are plotted here. Just to tell you, if you plot these different morphological parameters, you see that different types of creatures form clusters. For example, here in magenta, you have a cluster that is formed by bipedal dinosaurs, like T-Rex or something. And you have, for example, in yellow, another cluster that is gliding, soaring flight creatures. And now Archaeopteryx, if you look at it, is this little gray triangle down here, which is right in this cluster that is a little bit cyan in color. And if we look what this is, we see that it's animals capable of short flight. So this shows that Archaeopteryx has a lot of physical parameters that are actually very close to other animals that can also do flight. So with the knowledge we have nowadays, it's most probable that Archaeopteryx was indeed capable of powered flight. It means whenever he wanted, he could just flap his wings and take off. Of course, as we said, he doesn't have the same morphology as modern birds. So that means when he flew, it didn't look like a modern bird flying because he couldn't do the same wing stroke. But nevertheless, he was able to fly for, well, of course, we don't know how far, but at least for short periods of time. That's again a very fundamental change in the view of a species through X-ray diffraction tomography. Okay, we're going to finish with Neutron tomography. So Neutron tomography is very complementary to X-ray tomography. So we're typically again going to look at larger samples, more absorbing samples. So we typically have resolutions of around a millimeter or a fraction of a millimeter. And Neutron tomography is, of course, ideal to image light Z materials inside heavy metals, for example. And a very typical study I can show you here is this work that has been done on this lead sculpture. So this is a sculpture made out of lead that has been made on the top of a wooden scaffolding, which you cannot see, it's inside. And now the conservators wanted to know, but well, is the wood inside, is it still okay? But of course, there's no way to look into it. You cannot do a radiograph because, of course, the lead would block it. So they had to use neutrons, and they went to a neutron source, and you can see the result of the neutron tomography here, and you can see the reconstruction of the wooden scaffolding inside the statue here. And then with some image treatment, they could also see where there was detachment. So in red, there are areas where the lead is detached from the scaffold. And you can see that is, for example, the case up here. So where you can ask yourself, maybe it was never attached here actually because it's resting quite fine here on the head and on the shoulders. But maybe it's because the wood is starting to corrode or to somehow to get bad inside, and the conservator needs to do something about it. So this is, of course, a very valuable information for the conservator. Another example is here this Buddha figure. It's made of bronze, again, a very absorbing material, and it's known to be hollow because it's a sacred object, and there are sacred objects that have been placed inside. And again, with neutron tomography, you can look inside without having to destroy it or open it. And you can see here the tomogram where you see very nicely through the bronze outside of the Buddha, and you can see different objects that have been placed inside. So you can, for example, see here a prayer scroll and then some pouches and other sacred objects. Well, that was all I wanted to tell you. So as I said, I only have very small time, so I can really show you a very, very small window out of all the things that have been done in cultural heritage research. I had lots of other studies I would have loved to show you, but unfortunately I didn't have the time to show you. However, I still hope that this has been really interesting for you and not too tiring, and I wish you a very nice evening that you have now really merited.